So today's topic is nutrition in animals. It's not a very lengthy topic, and we will be able to do it in maximum 15-ish minutes. This is the digestive system of humans. Here is our mouth, teeth, tongue, esophagus, salivary glands, liver, stomach, gallbladder, pancreas, large intestine, small intestine, rectum, anus. So it all starts by ingestion, which is taking in food by means of hand and mouth is where the digestion begins. So the physical digestion happens in mouth through teeth. The molars and premolars, they grind the food and chew, and while chewing, they break down large food particles into smaller food particles, which increases the surface area to volume ratio for the action of the enzyme, which is amylase, which works when it comes to chemical digestion. Saliva containing salivary amylase breaks down starch into maltose at pH 7 and 37 degrees Celsius. Saliva is secreted by salivary glands. Then comes to esophagus. Here the tongue rolls the food into bolus, into the esophagus, and then in the esophagus before the bolus, our lumen constricts because of the contraction of inner circular muscles and relaxation of outer longitudinal muscles to push food along the esophagus. And after the bolus, lumen dilates because of relaxation of inner circular muscles and contraction of outer longitudinal muscles to push food along the esophagus. So in both the muscles are antagonistic, so they're working vice versa. Then we have our stomach. Physical digestion occurs because the stomach is a muscular bag made of smooth muscles. So it contracts and relaxes to mix food particles with the gastric juice, which is secreted by the gastric glands. The stomach further breaks down particles to increase surface area for the action of protease. In mouth, only digestion of starch occurs, and in stomach, only digestion of protein occurs. So basically, gastric juice secreted by the gastric glands contains HCl and proteases. Protease is an enzyme. HCl kills the bacteria to prevent infections, and it provides the optimum pH, which is 1 to 2 for the action of protease. Protease breaks down proteins into peptones and amino acids. Then comes the small intestine. The small intestine has two parts. One is the duodenum and the other one is ileum. The duodenum receives secretion from 3 glands. All 3 fats, protein, starch are digested here. First gland is liver. Liver secretes bile stored in the gallbladder and powered into duodenum through bile duct. It breaks down large fat particles into smaller fat particles to increase surface area for the action of lipases. This process is called emulsification, and bile is the only secretion in the digestive system which does not contain any enzyme in it. So bile is not an enzyme, it's just a secretion which emulsifies fat. And then pancreas. The pancreas secrete pancreatic juice containing pancreatic amylase, lipase, and protease. The amylase breaks down starch into maltose at alkaline pH. The duodenum contains all secretions in alkaline medium. So it breaks down starch into maltose at alkaline pH. The pancreatic lipase breaks down emulsified fat into fatty acids and glycerol. And the pancreatic protease, it is originally secreted in inactive form, and then it is activated and it breaks down proteins into peptones. And then we have intestinal glands. So they secrete intestinal juice, which contain intestinal carbohydrates, intestinal lipase, and intestinal protease. Intestinal carbohydrates have 3 enzymes: maltase, sucrase, lactase. Maltase breaks down maltose into glucose. Sucrase breaks down sucrose into glucose and fructose. Lactase breaks down lactose into glucose and galactose. Basically, any word you read in this chapter having the word ase in the end, like maltase, sucrase, lactase, the ase part, it's going to be an enzyme. And then we have intestinal lipase, which breaks down emulsified fats into fatty acids and glycerol. And then intestinal protease, which breaks down peptones into amino acids. By the time food comes to duodenum, most of the digestion takes place in duodenum, and then absorption takes place in ileum. What happens in the ileum is that absorption of the end product of digestion occurs in the villi of ileum. The villi are numerous finger-like fold projections which increase surface area for absorption. The epithelial cell lining in villi have even smaller villi, which are called microvilli, and they increase surface area. The microvilli secrete amylase, lipase, and protease one last time to complete any incomplete digestion of starch, fat, or protein, respectively. And then we have goblet cells in the villus, which secrete mucus to lubricate the villus epithelium for a quicker absorption in dissolved state. So all the end products, they're dissolved in the mucus, they're then absorbed. The villi have a rich network of blood capillaries, and glucose amino acids are absorbed into the blood capillaries, while the fatty acids and glycerol are absorbed into the lacteal duct. And then we're coming towards the end of digestion. Assimilation, what happens is that in the colon, in the large intestine, it absorbs all the water and moisture from the end product through osmosis, and minerals are taken up by diffusion, and then digestion occurs. The rectum stores our feces, and the anus is used for defecation. And fibers are not digested by the human body, so they are digested. Then we have a short topic on teeth. So there are 4 different types of teeth: premolars, molars, incisors, and canine. Every human has each 4 of them. And the upper part of the teeth is called the crown. And it is a white visible part covered with a hard substance called enamel. It protects the inner structures, and enamel gets stronger with fluoride ions. This part, which is invisible and inside the gum, is known as the root, and it is the innermost part of root and crown. And the part which lies inside both of them is called the pulp cavity, which contains the blood vessels and nerves. So each human has 32 teeth. Every adult has. We grow teeth and we break them too when we're young. The first type is incisors, which is this. They're broad and flat, chisel shaped, used for cutting and biting. Canine are pointed for gripping and piercing. Premolars and molars are pretty much similar, except that molars have triple root. They're wide with a central depression. Both are used for grinding. So we have 8 incisors, 4 in upper jaw, 4 in lower jaw. We have 4 canine teeth, 2 in upper jaw, 2 in lower jaw. Premolars, 4 in upper jaw, 4 in lower jaw. And molars, 6 in upper jaw and 6 in lower jaw. So incisors have a single root, canine have single root, premolars have double root, and molars have triple root. And then we have the topic tooth decay. Tooth decay is termed as the erosion of tooth due to bacteria buildup. Causes are the improper brushing, excessive sugary food intake, no dental visits, less fluoride intake. Prevention is the exact opposite, like regular brushing, less sugary food intake, regular dental visits, addition of fluoride in water. Could be in water, in diet, or in toothpaste. And we're done here. Thank you so much.